Physiology lecture 4 of exam 3, talking about absorption, intestinal absorption. Carbs are absorbed. There's 3 of them here. There's starch, lactose, and sucrose. Starch is by far the most abundant, comes from plants. Lactose comes from milk, and sucrose is also really common sugar. So starch is broken down by salivary amylase, which hydrolyzes it. Salivary amylase is released from the pancreas. I believe it's secretin that causes the release, but I'll double check that. So anyways, starch is broken down into maltose. And then on the cell membranes of the microvilli, there is something called maltase, which breaks down the maltose into more simple sugars. Lactose is broken down by lactase, and sucrose is broken down by sucrose. Pretty straightforward. Now going into proteins. Proteins, pepsin cuts the collagen. So pepsin is released from gastric chief cells in response to high levels of acid. The acid is secreted from the parietal cells in response to histamine, and histamine was released from the enterochromaffin cells in response to, I believe it is gastrin, and gastrin is released from the gastrin cells, the G cells, which are located in the pyloric. Can't think of the word. Anyway, all right. So this pepsin, it used to be pepsinogen. Its favorable pH is about 2 to 3. If it gets up to pH of 5, the pepsin doesn't work anymore. The pepsin is the only thing in our body that can cut collagen. Collagen is a really common thing found in meat. So the pepsin breaks this up. Then you got these polypeptides, and the polypeptides are broken down into dipeptides by proteases. These proteases are located on the membranes of the microvilli. So now the polypeptides are broken down into dipeptides. The dipeptides are broken down into amino acids, individual ones, inside the cell. These dipeptides get absorbed into the cell by these cytosolic peptidases. So the cytosolic peptidases break down the dipeptides into amino acids in the cell, and then these amino acids are then released into the blood, where they go where they need to go. Okay, now lipids. Lipids, we eat fat. It's the triglyceride, kind of the fat chunks into these triglyceride droplets, these big ones, large ones. They're emulsified, I believe it's through bile salts, into smaller droplets, and which the bile salts have like a positive end and a negative end, so it kind of acts a lot like salt really, and they kind of surround these droplets, kind of break them off, and they surround them into these really tiny things. And these small lipid droplets then are able, they kind of basically make it so that they have more surface area for the lipase to have action. Because the big one, it only has the outside, but lots of little ones, it's got lots of surface area. So then the lipase breaks down a triglyceride into a monoglyceride and 2 fatty acids. And these bile salts are then, recycled, but apparently these bile salts still form micelles around these products, and then these micelles, which contain the free fatty acids, the monoglyceride, they kind of go onto the microvilli, and then they go into the cell, and then for some reason the body reforms the components of the triglyceride, which are the 2 fatty acids and the monoglyceride. It seems like there'd be 3 fatty acids, but whatever. It's triglyceride. Okay, so then it reforms them back into a triglyceride, and then they are coated with proteins inside the cell. So now you've got a triglyceride again. They're coated in proteins, and the triglyceride coated in the proteins forms something called a chylomicron. The chylomicron is now released from the cell, and it is taken up by the lacteals, which is some little component in the microvilli, I believe. No, it looks like it's a component in the villi. Taken up by the lacteals, passes through the lymphatic duct into the subclavian vein, and then I guess it's dispersed from there. Let's talk for a moment about the surface area of how these things are absorbed. So if we're looking at the intestine here, we've got these big valvulae. Okay, these are circular folds. Increase the surface area by 3 times. They have other names too besides valvulae. Sometimes they're called rings or plicae circularis. But anyway, big things. Now on these big things are these, actually like this, these villi. Okay, so this is the big plicae the valvulae or plicae circularis or keck ring, whatever. On these are these villi. They're about 1 millimeter tall, and they're found in the intestines all the way from the pyloric sphincter all the way to the ileocecal valve. So basically, like the entire small intestine. They increase the surface area by 10 times. Okay, maybe it helps to think of it this way. So here's 3 different. Let me do it this way. Here's 3 different valvulae. Each valvulae has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 villi. Just pretend that's 10. So right there, we've already got this, like 30 times the surface area just by 3 of these things. And so these villi, each one contains an artery, a vein, obviously, because you gotta go in, gotta go back out, and capillaries, and also something called a lacteal. Okay? Each one of these, I did it inside the valvulae. It's actually, think of it inside of the villi. Okay? So an artery, a vein, capillaries, a lacteal inside each one little villi. Okay? And also, now we're going to go even smaller. On each one of these on each one of these villi are microvilli. So these are like just little tiny, I mean, they're like one I think it's micrometer tall, and they're only like 0.1 micrometer in diameter, and there's actually 1,000 of them per cell. So for every cell on here, there's 1,000 of these guys. They're on the epithelial cells. They increase the surface area by 20 times. So altogether, 3 times increased surface area by these plicae circularis, 10 times the surface area by the villi, and now 20 times by the microvilli. 3 times 10 times 20 is 600. So altogether, surface area is increased 600 times. That's a big deal. This allows for a lot more absorption. So there's other things that are absorbed too. On these microvilli or villi or whatever, you need to create a favorable gradient for absorption. So the way that this is done is the sodium-potassium pump pumps sodium out, into this little area where the microvilli are. And that sodium now is going to create a favorable gradient for the transport of amino acids, glucose, and galactose. Without sodium there, apparently these things wouldn't be able to come in. It also creates a favorable transport for chlorine as well, and water. So I mean, the sodium-potassium pump's doing a lot here. Note though, fructose actually just goes through, so I said glucose and galactose, the 2 G's, they need sodium, but fructose actually just passes through by facilitated diffusion without sodium. So that's an interesting note. Now, iron. Let's talk about iron for a second. So iron, we need iron in our body. Obviously our blood has iron. So how do we get the iron? How do we absorb it? Well, our liver secretes something called apotransferrin, which goes into our bile and comes out in our bile. And this apotransferrin binds free iron. It binds hemoglobin and myoglobin. And when apotransferrin is bound to these, iron or whatever, or hemoglobin. It makes something called transferrin. And then the transferrin basically allows it to go into our body, but it makes sure free iron is actually not good to have in our body. So this binds it up and takes care of it, so we can absorb it. So also chlorine, HCO3, and calcium. Chlorine, how does our body absorb chlorine? It diffuses into our bodies because of the sodium gradient established I talked about over here. So because of the sodium gradient, the sodium's going out, it allows the diffusion of chlorine to come into the cell. The ileum and large intestine absorption exchange for HCO3. So also, if you're looking at the ileum and the large intestine, HCO3 is exchanged for chlorine. So they're going to swap it. The HCO3 is going to go out, the chlorine is going to come in, I guess. I'm assuming that's direction. Now I'm talking about HCO3. In the upper small intestine, hydrogen ions are exchanged for sodium ions. When you combine the H, so the sodium goes out, the hydrogen comes in. Then the hydrogen is combined with HCO3 to form CO2 and water. And now calcium just absorbs in, probably from the favorable concentrated gradient from the sodium as well, but for its absorption, vitamin D and parathyroid hormone are also necessary. And lastly, water. Basically it's osmosis. All the salts in there, or sodium, the ions, whatever, you basically absorb all the water. You can absorb 5 liters of water or something like that. And that the sodium, the water is not allowed back out because of tight junctions. And that is everything from the lecture.